Hi, so today we are going to try to understand gram-negative bacteria. As we remember from the last video that we did, that gram-negative bacteria, they do not retain the stain, right? And that is why they are called gram-negative, and they generally take the counter stain, that is your safranine, right? Safranine is the counter stain, and that is what they take up because of the thin uh, peptidoglycan cell wall. They allow the stain to be washed off. I hope you remember that the two kind of bacteria, negative and positive, are because of differential staining. Positive has a huge thicker cell wall above the plasma membrane, whereas the negative has a thinner, right, a thinner peptidoglycan layer and an outer membrane. Because it has a thinner peptidoglycan layer, that is why alcohol, the mod first, the iodine and the crystal violet complex, it doesn't fix very much, and alcohol is easily able to rid some outer membrane, wash out some outer membrane, and hence during the decolorization step a lot of stain is washed out. But even though it does not stain well, it has a lot of other things that we need to understand. Now, also you would like to know that the thickness of this layer is very less. That is why it is there, right? So this outer membrane is actually not very thick, but yeah, a little bit more than the peptidoglycan layer, which is very thin. Now, this space called periplasmic space between the two outer membrane and the plasma membrane. This space carries a lot of significance as well for us. Now we'll again start with the peptidoglycan. We already know from the other video that it has a carbohydrate derivative, that is NAM or NAG, right? But the thing gets really interesting when we start talking about the amino acid chains. Now, I hope you remember that we had 5 in gram positive bacteria, 5 glycine peptide linkages between the peptide short chains. But here there are two differences. First is the difference between the third amino acid. If you remember, in case of gram positive, that was lysine or mesodiaminopalmilic acid, but here we have DAP or diamino pymalic acid, right? So this is the one that we find too much here. Okay, now what about the linkages? So the third DAP of one side and the third DAP of the other side of the strand, they are linked here, right? So there's a difference in the linkages as well that we can clearly see and find in the structure. Now, let us talk a little bit about the negative cell wall. So this is obviously the plasma membrane and a very thinner peptidoglycan wall, and just above that we have an outer membrane. Now, very interestingly, we should understand that when we refer to the cell wall, actually both the outer membrane and the peptidoglycan layer, they are both together called as the cell wall. In what else? Now, there is a periplasmic space. What is the periplasmic space? That is the space in between the two membranes, right? So this is the periplasmic space. Now this space has a lot of hydrolyzing enzymes. So these enzymes perform hydrolysis of a lot of substances. Now I hope you this makes only sense because a lot of gram negative bacteria are actually pathogenic, right? So this feature comes in handy. Now there's also a lot of transport proteins which allows them to get hold of a lot of nutrients allows a lot of nutrient intake for the gram-negative bacteria. So this is the periplasmic, uh, the proteins which are next to the periplasmic space, and the outer membrane also has got some lipopolysaccharides. Now these lipopolysaccharides can have different functions. Sometime some of the part of the lipopolysaccharides, they are toxin. Okay, so they are toxic, and they are toxins to the other cells. Now, let's try to understand the various components of, or various functions of this lipopolysaccharides in a little bit more detail. So these lipopolysaccharides, as I said, sometimes are toxin in nature. Besides, they also contribute to the negative charge of the outer membrane. They also control the permeability. A major component of the outer membrane is these lipopolysaccharides. So that is why they are able to control a lot about the permeability as well. They also protect the bacteria. So they also have a role in protecting the bacteria from host defenses. So they protect the bacteria. So as you can see, these polysaccharides have got a lot more function. Now a very important component is the bronze lipopolysaccharide. Now a bronze lipoprotein is of another importance to us, because this bronze lipoprotein covalently links, so it covalently links the outer membrane to the plasma membrane, and thus is of a greater significance. So we see, even though the gram negative walls are a little bit thinner, it has got a lot of other machinery that makes up for it. I hope this makes sense.
We hope this video was helpful. To see more biology topics covered, click on our channel and don't forget to subscribe.